And now, an exclusive sneak preview of a recent X-Talks webinar, Cryopreservation Best Practices for Cell and Gene Therapy Source Material.
Why do we cryopreserve cellular and gene therapy products? The main reason is to make these cells ready for patients when the patients are ready, particularly for autologous products like for bone marrow transplant following high-dose chemotherapy or the manufacture of CAR T cells. The cells may be collected from the patient long before the patient is ready to receive the treatment. Cryopreservation ensures that the therapy is safely stored for an extended period of time until the patient is ready for the administration of the treatment. Oftentimes, the donor and the recipient are long distances from each other. Cryopreservation of the cells makes these cells available to patients all over the world from donors that are also all over the world. Another major factor for cryopreservation is the ability to localize where these cell materials are manufactured. They can be shipped from one place to the next, and as I've already mentioned, we ship throughout the world. Next slide, please. Cryopreservation is a critical step in the complex processing and manufacturing of cellular material. Different biomaterials or different cell types require different preservation techniques. The cells are prepared by freezing based on the unique properties of the cells and their tolerance for the freeze-thaw cycle. The success of the procedure depends on skillful preparation of source material during processing and following all appropriate protocols to suit the cells of interest. And these protocols may differ from cell type to cell type. Red blood cell and plasma reduction is often required for hematopoietic progenitor cell products in order to ensure that the red cells don't lyse and cause all kinds of problems for patients undergoing treatment later on. A cryoprotectant, which is a chemical that preserves the cells during the cooling process, is added. Most frequently, it's dimethyl sulfoxide or DMSO, but more complicated and extensively manufactured cell therapies result in more fragile cells, and sometimes a DMSO alternative is necessary. Freezing can be done by either controlled rate freezing in a computer managed freezer that reduces the temperature in the chamber gradually according to a cell program, or passively by simply putting the cells into a very cold freezer and allowing them to cool down gradually on their own. Long-term storage can also be in either vapor or liquid phase of liquid nitrogen, and the temperatures of each of these are somewhat different, liquid phase being the coldest. Cryoprotectants such as DMSO and non-DMSO alternatives result in altering the fluidity and permeability of the cell membrane. As a result, they influence whether or not water flows into the cell or out of the cell. They prevent the cells from changing their tonicity and end up with either hypotonic or hypertonic environment. This preserves the cells by preventing lysins during the cooling and thawing. Pre-freeze and post-thaw exposure needs to be limited. Either one can result in lysis of the cells. And generally, the thawed products are infused immediately upon thawing. The DMSO infusion dose should be limited and is generally considered safe at less than 1 gram per kilogram per 24 hours in order to reduce adverse events. 1 ml of DMSO is equal to 1 gram of DMSO, so it's pretty easy to limit that dose. The effects of non-DMSO alternatives are still being investigated as these different materials are being developed for their purpose. Next slide, please. Umbilical cord blood is one of the major sources of hematopoietic stem cells, and they have been used in bone marrow transplants since 1989. I'm going to turn this program over to my colleague Ayanna Bryan right now, and she will take over.
To view the entirety of this webinar, Visit the link in the description and register on xtalks.com completely free of charge.